I'm already live. Okay, will it be frowned upon if I take a sip of my coffee? I've had so much fun with my email. I tell you what, I don't have internet in my office. I made it here. I'm getting a talk this afternoon and I don't know what computer I'm doing. I haven't finished writing this bloody talk. So anyway, if nothing else, I'll give the same talk. Can people hear me okay through the mask and stuff? Is my microphone on? No? Hello, hello? Should I hold it up? It's not a bad idea. Is this better? I don't even know which side it's on now. Okay, can people hear me okay in the back or in the front? Yeah, we can't start until I'm on Zoom. So ask me your embarrassing questions now. Oh. I have an echo. Should I stand somewhere else? Where's the, it's coming out of that? Is this better? No, I hear it. I hear the echo. I got to tell you, it's funny. I don't think I've given a talk in about 2 years in front of an actual audience instead of in my garage. So do I have a remote clicker? Otherwise I can stand here. Oh, perfect.
So we're running late, and I completely forgot about the fact that someone needed to introduce Anna. So that's going to be me, and I'm going to tell you that Anna Dornhaus is a very beloved and amazing member of our community who does incredible behavior work with ants and bees, and we're so happy to have her talking today.
Thank you very much for that sweet introduction. So I'm trying something new with the talk today, so bear with me with that. I'm sort of mixing, I have this smattering of different results that I wanted to present, and I'm mixing it a little bit with some thoughts about philosophy of science, which probably most of you don't normally talk about or think about. Let me just click this away. Can I? No, I can't. Okay, anyway, so so thanks for coming to this talk with this kind of weird title, and there will be data on bees and actually data on humans, although very little. Okay, and this is sort of my outline, but I don't want you to read it right now. It's too long and complicated, but I'll walk through it as the talk goes on. And this is the more overarching outline of the points I want to make. I want to talk about exploration versus exploitation, and I'll explain what that is, but some of you are probably familiar with that phrase. And then I want to talk about the role of innate bias, possibly in decision-making in that exploration-exploitation context. And then in the think about the why, it's about you. So I want everyone to think more about why they're doing things the way that they are doing them. And I'm somehow going to try to connect what I'm going to say about bees to what you do in your life to find information and learn things. And I'm talking about how bees learn things as well. Okay, so if you'll bear with me, I'll explain how that's all connected. Okay, so the first thing to realize, and it's actually, we as scientists sort of pay lip service to it, but I think in our gut, it is incredibly hard to accept the fact that life is uncertain, that no matter how much we learn, we cannot perfectly predict the outcome of any of our actions. And there are some very clear reasons for that. And it's not necessarily that the world is stochastic at its core. That's sort of a philosophy/physics question, whether atoms and stuff actually have inherent randomness. Even if the world were deterministic and atoms didn't have any stochasticity at all, the fact of the matter is that the world is incredibly complex. By that I mean every outcome that we care about, but also a lot of outcomes we don't care about that seem trivial, are actually influenced by so many factors that it's impossible to know all of them. And it's also true, this is sort of a result from chaos theory, but it's a very general result, that in many systems, especially when many factors interact, small differences in input can lead to huge differences in output. And the most straightforward example is when you roll dice, right? There is a reason why they come up the way they come up. There's wind, and there's exactly the angle with which you roll them, and the forces, and so on. But the fact of the matter is just a slight change in angle can already lead to a totally different number on the dice when you roll them. And so to all intents and purposes, it seems stochastic because it will be impossible to measure all the factors that go into which side comes up with sufficient precision, precisely because small differences will lead to huge differences in outcome. And I really recommend Joanna's talk called There Is No Certainty. It was on one of the public lecture series on this general topic of there is no certainty. So any attempt to learn anything has to deal with this fact that there is no 100% certainty, that there's always noise. What we describe as noise is just a phrase to say, well, we can't predict exactly every outcome. But despite the fact that there is noise, there are also patterns. And so now turning to bees, when we talk about bees, bees need to collect pollen and nectar. I'm mostly going to talk about nectar foraging. And so I'm talking about generalist bees like honeybees and bumblebees. All my data will be on bumblebees. When bumblebees are faced with a scene like this, what the bumblebee would like to do is to find the flowers that have the most nectar per time invested, or something like that. But there's an incredible array of different plants. Some belong to different species, and the species differ in nectar production. But also even just subtle differences in the exact positioning of the individual plants may be associated with slightly different water availability, or different shade and sun, or different temperature if you're going over the crest of a hill. So like the dice, there are a lot of factors that will affect how much nectar exactly is in each flower. So the signal that the bees are getting when they're trying to find out which is the best flower is incredibly noisy. It's also noisy because other bees and other pollinators will empty some flowers and so on. So there's a lot of unpredictability in the detail, but there are patterns on the average. And the central problem of bee foraging is, how can a bee, given this noisy input, learn anything about what the average long-term pattern is, right? If the bee wants to find out, are the red flowers better or the yellow ones, or whatever set of different species there are, or different locations, the bee wants to extract the pattern from this very noisy input. So what strategy can a bee use to make a best guess about what that average pattern is? So for example, to determine the best flower species. And this is an inductive problem. So this is my philosophy slide. And there's a couple more later, but this is just to introduce that term. So in philosophy of science in general, we talk about two processes of gaining knowledge or making claims. One is deductive and one is inductive. So deduction is when you know what the general principle is and you want to find out something about specific cases. What I said in the beginning is we can never know the exact value of a specific case, but if we already know the general principle, then we can derive something about specific cases absent the noise. The other direction is much more problematic. If we have a bunch of specific measurements and we want to derive the general principle, that's what we call induction. And that is inherently incredibly difficult. In fact, there is no process to do this with absolute certainty, no matter what the level of noise. And most of science is like that, right? In science, we are trying to extract general principles from these individual measurements we're making. And that's also what the bees need to do. The bees want to derive a general principle about whether the red flowers are better than the yellow ones from individual measurements of a few flowers that they're making. So it's a similar process. And that similarity is what I want you to keep in mind as we're talking about bees. Okay, so in science, how we do strong inference, how we do induction. So how we do this from going from specific cases to general principles. There are two broad principles that we're using, and we're not going into too much detail in each of them, but I just want you to have heard the terms. So one term is strong inference, which is what we sometimes call the scientific method, although that term has a lot of misunderstandings. But the key point about strong inference is, you've probably heard this, you have a question and you think about alternative hypotheses before you look at the data. These alternative hypotheses make alternative predictions, which are specific statements about what the data will look like if this is true or if this is true. And then you make measurements and you compare those to the predictions. An alternative method, though, is called Bayesian reasoning. And Bayesian reasoning is you have some prior expectation, wherever that comes from, about what is true. And each time you get new data, you kind of include that, you use that new data to update your expectation about what's true. So this is kind of an instant all or nothing test. Whereas this is kind of a continuous process where you get new data all the time, and every time you update what your prejudices basically were a little bit. And of course, you could emphasize one or the other more and so on. But the principle of it is that every little bit of data you get updates a little bit what you think is true. Okay, and so in general, for Bayesian reasoning, the emphasis is on these priors, right? Bayesian reasoning is about including your prior expectation. This is a cartoon from XKCD. Crack, boom, there's a thunderstorm, there's two hikers here. One of them says, whoa, we should get inside. The other one says, it's okay, lightning only kills about 45 Americans a year, so the chances of dying are only 1 in 7 million. Let's go on. And then the caption reads, the annual death rate among people who know that statistic is 1 in 6. So the joke here is that, of course, this might be the overall rate, but you should, so this is your prior expectation, but you should be updating with the new data that you're actually very close to a lightning storm, and your probability may be different than that. But the point is that I want to make in every, so XKCD has multiple cartoons about this, about this topic. The point is Bayesian reasoning is always combining your background expectation with the actual current data that you have. And in fact, we think that bees are doing exactly that, and that is what I'm going to present some data on. And I think that this framework of Bayesian reasoning is basically captured by these two terms of exploitation and exploration. The terms are usually used in a slightly different context, but what I'm saying is exploitation means you're using the information you already have. So wherever it came from, you're basically using these priors. You're not trying to update with new data, you're just using the information you have and you're acting on it. Whereas exploration, what we term exploration, is the process of trying to acquire more new data in order to maybe get a better updated view of the world. Okay, so here's a bee trying to decide whether to stick with the flower it knows or look at new flowers. So this is what I just said. You could use prior individual experiences, or you may have innate biases, which is another form of a prior. It's prior knowledge acquired through an evolutionary process, versus collecting new data, meaning going to flowers that the bee doesn't know anything about yet, or not sufficiently much. And what that looks like, what exploration looks like, is that the bee goes to something that isn't its current best guess about where the best nectar is, right? So exploration, by definition, is something that what in a learning experiment we would call an error, right? It's when you're looking for new information, that means you're not just going to the thing that you think is the best thing to do right now. You're trying to explore other things that you don't know yet whether they're good, or even maybe you've previously visited them, but you think they might have changed. And so in order to keep your worldview updated, you have to go back to them. So that's exploration. And this is what I want to talk about right now. And I want to talk a little bit about, well, how can we tell if it's not errors? Because of course, they're bound to make errors, even if evolution had not, even if there was no reason for bees to do exploration, they didn't want to do exploration, there's bound to be noisiness in their decisions anyway. So how can we distinguish actual errors from an actual attempt at acquiring information? Okay, so this is the exploration exploitation point that I want to talk about first, and this is really the meat of the talk. Okay, so if we think that bees are combining expectations, these priors, with current data, why don't they just use all current data? Current data seems better than whatever priors you have from whatever source. And how much should you weigh those things? Where do the expectations even come from? And I have to warn you that I'm not giving comprehensive answers to these questions, but I'm just showing you some data on each of these points. And one of the overarching points that I want to make is that this exploration is actually pretty costly. Okay, so now let's turn to the actual bees. So here are bumblebees, Bombus impatiens. This is what we have in the lab. Most of the studies I'm going to, in fact, yeah, all of the studies are on lab bees. And this is not always what we use, but this is an example of what we use as artificial flowers. They're just little foam discs, and they have a little well that offers sugar solution for the bees to drink. These ones are from Shannon McWater's experiment, but I'm showing data from a variety of people. Okay, so the first point I want to make is that bees actually also live in this world where everything is uncertain. There's a lot of uncertainty, even in their perception about color and about how much nectar there is. And for this one, I'm not going to go into detail how the study actually worked, but I just want to show you the result that shows that there's uncertainty. So the bees here are foraging on this array. That's a vertical array. The study was done by Annie Leonard in Dan Papich's lab in collaboration with me. And so the bees are confronted with this wide array of colors that are sort of blending into each other that are pretty similar. Before they're confronted with the array, they're trained on these very similar colors. So they're trained to this greenish blue being positive and the more blue being a negative stimulus. And so in this case, the bees are trying to avoid the blue and they're trying to find the greenish blue. But what they are doing is when the blue is actually costly, they're shifting their preference. So this is the preferences of the bees. And this is how much they prefer each of these colors that are here on the spectrum on the bottom. And in one case, the red one, they are exactly preferring this color. Notice that they do make a lot of errors, if you want to call it that. They do go to all the other colors some. But then if the errors are more costly, if the blue one is actually negative, they end up preferring something. The positive stimulus is the same thing, but they end up preferring something that's removed from the negative stimulus. This is called peak shift. This phenomenon is pretty well known generally in other organisms. And it's called peak shift because the peak shifts. And the interesting thing is, so they prefer something that they were not trained on. They prefer a color that's actually not the color they're trained is the positive stimulus. And why is that? The theory is that they do this because they want to reduce the number of false landings on this negative stimulus. So this is their goal, going from this red line to the orange line down here. They want to reduce the number of costly mistakes they make. And they reduce the number of costly mistakes by, when in doubt, going to the greener color. And that ends up shifting them to a green color that isn't even the one that they're trained on. Okay. So that just shows that they're uncertain about the color, uncertain enough to choose a color they weren't even trained on. But even more to emphasize the point that this is about uncertainty, if you add a second type of stimulus, so in this case, there's an odor associated with the flowers. Interestingly, the odor doesn't allow to discriminate, but in any case, the point is there's another source of information there. And with this other source of information, they make fewer mistakes and no peak shift occurs. So this peak shift, that's a sign of uncertainty about the color, disappears when they also have scent or odor to guide their choices. Okay, so this was just to make the point that bees can even be uncertain about the color that they're seeing and whether or not it is the color that they remember. Bees also invest time in learning. So this is all about this point of exploration, and exploration being pretty costly. So this is actually a pretty old experiment. This is one that I did in my PhD. The bumblebee colony was here, the bumblebee was flying in here and then through this hole. And once it flew through the hole, it was confronted with an array of flowers. These were actually projected on a screen, but whatever they were, and this is also Bombus terrestre, so it's a different species than the one before, but it's still bumblebees. Okay, so the bumblebee is making choices. And again, the bee is trained to prefer one color over another color, and the colors are pretty similar. So the bees are uncertain about which one is the right one. And the data we found there, so this thing with the hole is just so that we could actually measure the time from when the bee first could see the flowers to when the bees was about to land on them. So we can measure something that included the decision time, the time to think about where to land. And what we found was that the decision time correlated across different bees, so each point is a different bee, with the percentage of correct choices. So some bees make fast decisions, but they make fewer correct choices, and other bees make slow decisions, and they make more correct choices. So it seems that there's a difference between bees in their tendency to prefer fast decisions, which may be erroneous, to slow decisions, which are more accurate. Now, this could just be that the bees have, it's unclear what causes the bees to be different on this dimension. But the interesting part of this experiment is that they can tune that decision, that trade-off between speed and accuracy. The bees are making an active decision about where to be on that. And we showed that by making the errors more costly. So again, in this case, we made the errors more costly by adding quinine to the negative stimulus. So instead of just having water, which is uninteresting, they actually have this bitter solution that they really don't like. And what all the bees end up doing, every single one of these individuals increases their accuracy when errors are more costly. And they all also increase their average time to make these decisions. So in other words, all of the bees, when their errors are more costly, make the decision to invest more time and they get more accuracy out of it. But the ordering of bees is more or less preserved. Okay, so if you think of this in this framework of exploration, of having to do exploration in order to get new data and what the world is like, the exploration is costly. Actually trying to learn and take into account this new information on which color is worth it is taking extra time. Okay, and then lastly, this is really new data. So this is from a study that was led by J.M. Mazel in collaboration with Bob Wilson in psychology. We were examining something that Bob calls directed exploration. So this is something that Bob Wilson is studying in humans. And it turns out directed exploration in humans is associated with distinct levels of dopamine and all kinds of sort of hard measurable differences between people in between situations. And so what we mean by directed exploration is when bees have a choice between an option that's more informative and an option that's less informative, they actually prefer the more informative option. And to explain how we found that, we did, I'm going to show a photograph later, but we had this repeated Y maze. So we basically make the bumblebee walk through a very long tunnel that repeatedly splits and splits and splits again. And so at each split, the bee is making a choice between left and right, and there's a color associated with it. And so at each decision, the bee is choosing one color and gets a reward or doesn't get a reward, or gets a reward that differs in magnitude. And in this case, we first force a few choices. So what this is supposed to indicate is that one arm is blocked off. So for example, in this picture, it's an example. The bee is forced to get to the red flower, and then blue flower, and then red flower, and then so for these first 4 choices, the bee has 3 experiences with a red flower and just 1 experience with a blue flower. And the amount of nectar varies a little bit every time. So the bee is now trying to guess, on average, is the red flower better or the blue flower better? But for the blue flower, it only has 1 sample. For the red flower, it has 3 samples. So regardless of what the difference in average is, the bee knows that it has less information about the blue flower. And so on the next choice, where the bee actually has a choice between blue and red, the blue is the more informative option. Does that make sense? Because the blue is the one that it knows less about, and the red is the one that it knows more about. And so bees do prefer whatever has a higher average, but statistically, when you examine their choices, they prefer the more, if you extract the effect of higher average yield, they prefer the more informative option. So they care both about their current knowledge of which one is higher average, and they care about which one is more informative, and they choose the one that's more informative. In other words, the one that they have less experience with. And you can see that here by the fact that both of them are above 50%. We also had 2 conditions, one in which the bees were allowed to make 1 choice and one in which they are allowed to make 6 choices. In theory, you would expect that if you know that you have 6 choices ahead of you, information is more important for this choice, right? Because then you'll get to use that information 5 more times. But we didn't find a significant effect of that. So there's no significant effect between the 2. There may be a power issue, or maybe that bees don't care about horizon, or maybe that they don't believe us that they're really only going to have that 1 choice. So we don't know that. But what we do know is that in either case, they prefer the more informative option. Okay, so that's evidence for deliberate exploration, or what Ball calls directed exploration. Okay, so what I've argued is that bees can be uncertain about color and rewards. They do invest time in data collection, meaning collecting new evidence, going to new flowers and learning. They prefer informative options, and they can tune the amount of exploration they're doing to the cost of errors. Okay, but do they actually integrate some kind of prior with the new data? Do they actually mix these 2 information sources? Well, there's actually lots of studies that show that, but I want to show one that we have done recently, which this looks a little weird because it's filmed with a GoPro, so it's distorted. It's a flat arena bottom. And bees are given choices between blue and yellow flowers, and it's all controlled for blue and yellow. Okay, so we think that the innate color preference that bees have for blue has the role of a prior, right? It's like a prior information that they've acquired evolutionarily. They prefer blue. There's some evidence that this has to do with the fact that blue flowers on average seem to contain more nectar. We didn't measure that here. Okay, but it's well known that bees have a preference for blue. Okay, and in this experiment, we trained bees either on blue or yellow. And then after we trained them on one of the colors, we added the other color, or we added some other distractor, some other novel stimulus. And we essentially measured how much time do bees spend exploring this new thing that has suddenly appeared in their environment. Okay, so this doesn't have any, neither of them have nectar actually, but this is feathers or leaves or something. So there's not really any reason to expect that it contains anything. But the bees still explore new stimuli, and that's a part of their exploration. Okay, and it turns out that the time bees add when this happens and the time bees add when this happens correlates. So some bees explore more and they spend more extra time in either condition, and some bees explore less and they are less distracted by either type of stimulus. But what happens is the bees that were trained on blue generally don't spend a lot of extra time exploring whatever new thing has happened. Whereas the bees that were trained on yellow here spend a lot of time exploring the new stuff. And the way we interpret that is that the blue is their prior right? And when they have a prior that agrees with the new data they got in this condition where they were trained. So in other words, if they're trained on blue, they already expect blue to be good. Now they have experience, new experience that blue is in fact good. Now they decide they don't need any more new exploration and they don't care what we add to the arena. Whereas if we train them on yellow, then their recent experience and their older priors, the blue bias that they have, conflict with each other. And so they have a high tendency to spend additional time exploring the new stimuli in the arena. So if they're trained on yellow, that's these yellow points. Either axis here is the time, the additional time that bees spend when new stimuli are added. So when they're trained on yellow, they spend more time exploring, presumably because their original prior and the new information from here conflict. So they're trying to collect even more new information because their certainty is still low. Okay, and I just want to mention, without going to the details of the studies, there are actually lots of studies that show this integration between innate biases and current information that bees are collecting, where the innate biases really change how bees interact with new information and how certain they are about what they've learned. So Dan Papich, for example, has nothing to do with me, but he, with prior postdoc, had shown that bees pay a lot of attention to social information when you train them on a flower that they don't innately prefer. Whereas if you train them on a flower that they do innately prefer, they basically don't care about new information, social or otherwise, that agrees with our result. And David Kikuchi in my lab did a study that was different again, that had flowers that had a multitude of different signal components. In this case, they had a combination of different colors. And if one of the colors is one that they prefer, then they basically ignore the other dimensions of that signal. So once the prior, the innate bias, and the data they just gathered agree, they don't care about new information. It all goes towards the same conclusion. Okay, so this was my argument that bees in fact do exploration and exploitation, that exploration is costly, but that the data from exploration, the new data, are combined both with prior individual experience and with priors that come from innate biases. Now, okay, so why don't you use all current data? Because current data is costly to collect. I want to say more about that, and I want to say a little bit more about how they change how much exploration versus exploitation they're doing. So this is just a reminder. A lot of you probably know that, well, first of all, in social bees, it's only a small fraction of the colony that's even collecting food. The relevance of that is that if it's only 5% of the bees collecting food at all, they have to collect food for all the other bees. So they have to collect 20 times as much food in a day as a solitary bee has to collect, if we assume the total is the same, which probably isn't. So we're talking hundreds and thousands of flowers and decisions every day. They have to fly very large distances. There's a very high mortality risk during foraging. So this is all to say that if you want to do exploration, you want to collect data about this world, there's a lot of stuff to sample. It might be quite costly to collect all this information. And there's obviously a trade-off between the benefits of updating the knowledge that you have by doing exploration versus the costs of collecting that new information, right? So you could just stick to your priors. For example, you could just use your innate biases and not learn any new information. That would give you maybe a fairly good guess as to what the best thing is to do. But if the world changes, that means your priors become out of date. And so bees have to trade off this updating versus not updating. Let me make sure I have a clock here somewhere. Okay, so how do bees decide? And again, this is another study that we did recently on bees with actually an undergrad, Jack Kearsley, and David Kikuchi and Tim Polnashek were both postdocs in my lab. That shows, I think, another type of cost of exploration, which is the fact that bees actually have quite limited attention. So in this study, we had bees foraging on these 4 yellow flowers. And then at the end, we had them foraging on a novel type of flower, which honestly does not look that difficult, but is pretty difficult for the bees. So these are the novel flowers we had. So there's still these foam discs, but here there's some obstacles. Here they have to squeeze into the middle. Here there's a little cap. The cap of an Eppendorf tube is actually on top of the nectar well. And the bees have to push it aside to reach the nectar. And in this case, the cap is actually slightly pressed into the well. So they have to use more force. I see people laughing. So bumblebees are actually quite strong, and they are absolutely capable of pulling it out. But they have to figure out that that's what they have to do. So these were our complex novel flowers. And then in this case, we have novel flowers that are simply another color. In this case, there's just the trained flowers. And all of these setups are either in a simple environment or a complex environment. And long story short, I'm not presenting all the details of this study, which was on innovation. And the interesting thing about innovation in this case is that we show that innovation, meaning solving these novel problems, was easier for bees when they were in a simple environment than a complex environment. We actually weren't sure which direction it would go, which is good. Beforehand, we thought, and the literature says that in some cases, you kind of get into too much of a routine if your environment is simple, and that a simple environment can reduce innovation. But we found the opposite. We found that the simple environment increases innovation. And we think this is because of the attentional costs. So we think that in these environments, it's so hard for the bees to even focus on the thing that they want to do, namely finding the yellow flowers that they're already trained on, that it's very hard for them to do anything novel. Okay, so in this, just to explain what the results are, so the results show how many flowers are left over. This is survivorship analysis, and it shows how many flowers are left over. So how many flowers have they collected nectar from in the complex environment and the simple environment. And in the simple environment, in all the different conditions we had, they were faster at extracting the nectar. Okay, so my point here is that bees are more able to detect new flowers and innovate solutions in simple environments. Okay, so exploration is costly, data collection is costly, in both because the world is complex and large and there's so many things to collect data about, and because the data is so noisy. And this is exactly why innate strategies are actually so common across the animal kingdom. Because an innate strategy means you just have priors and you give up on updating them. And you just stick to that and you save all your exploration costs. But at the same time, if you live in an environment that dynamically changes, there are benefits to doing exploration. And so now finally, I'm going to show you cases where bees regulate the amount of exploration they're doing. So here's a photograph of that repeated Y maze that I told you about earlier. So in this picture, it doesn't have a lid. In the experiment, there's a lid on top, so the bees cannot get out. And they're forced to walk. This bee isn't actually in the photo, but this is about the right size. And the bees walk in this repeated Y maze, and they choose between blue and yellow over and over again. And we actually make them do about 100 choices in a row. And this is necessary because we want them to actually do quite a lot of sampling, a lot of exploration, in order to find out not just what is the average level for blue and the average level for yellow, but in this case, we're actually making the environment change over time. And in different sets of the experiment, we have the environment change at different rates. So in some cases, so there's always one is higher than the other. So let's say blue is better than yellow. And in the constant environment, for 100 choices, blue is better than yellow. But in the variable environment, let's say every 7 choices or every 10 choices, it switches. And yellow becomes better than blue. And we don't do it deterministically, so there's a probability of it switching. So it's not exactly every 7 choices, it's with a certain probability per choice. So that's what I mean by environmental persistence. And we find that they do what theory would expect, which is that bees explore more when the environment changes more. So here on the x-axis, we have the environmental persistence. More persistence means There's a 99% chance on every choice that it stays the same. Less persistence means there's a 60% chance on every choice that it stays the same, and a 40% chance of it changing. By it, I mean whether blue or yellow is better. And then here we have the proportion of choices that we call sampling. So that means choosing the flower that is not currently the best one. And we see that sampling goes down the more constant the environment is. So bees actually measure the overall rate of change in their environment and adjust how much sampling and how much new data acquisition they do, how much exploration they do, according to how fast the environment changes. Okay, so current data are hard to get, exploration is costly. Very briefly, you've probably seen me say this before, and I'm almost out of time, so I'm actually not going to go into detail on this, but I'll just say that social signals generally also reduce exploration. And just to give you the intuition for that, if you're doing what somebody else tells you and you're an ant colony, then everybody else soon ends up doing the same thing, right? The pheromone trail gets stronger and stronger to the one thing that's being recruited for because of the positive feedback. And so less and less people actually explore novel and distinct options, right? So generally recruitment or social signaling, social communication reduces diversity, reduces exploration. That's a very general result that is well studied a lot in ants. And we also show the same thing for bees. So don't worry about the data slide, but just admire these nice pictures of pollen grains. So Martina Donson Matashi in my lab measured honeybee foraging with and without waggle dance communication. And we also quantified what species, or at least what genera, they collected pollen from. And this is a summary of the result, which is that when communication is intact, if you think of the orange circle as all the available plants, and the yellow circles as what is one bee foraging from, all the different bees have very high overlap. So they pretty much forage from the same things. Whereas if there's no communication, individual bees overlap less, and so they're exploring more of the total of the environment. Okay, and we have data from ants that also, well, they don't show the same thing, but they're similarly ant species, different, in this case it's different species, differ in how strongly they adhere to recruitment. In other words, how many of the foragers actually follow pheromone trails versus doing their own thing. So that's measured on the y-axis, and it turns out that that correlates with their dominance ranking in the habitat. And it's probably, so this is a little bit an indirect argument, but essentially the fire ants focus more on dominance. They explore less, but they concentrate all their foragers on the same resource so they can be competitively dominant, but it means that they're sacrificing their ability to explore lots of different resources, which is what these Dory mimics are doing. Okay, and now I want to give you one last bit of data, but you still have time. And I've saved the best for last in the sense that I think this is really at least new to me and new to insect learning, and therefore I think really interesting. So we tend to think of innate biases as this rigid thing that prevents you from learning anything. But if you think of innate biases as priors in a Bayesian framework, that's not true. Innate biases are not mutually exclusive with learning and being updated. And the point that I'm going to make now is that innate biases can actually help you learn something if the initial phase of the learning is costly. So the argument goes like this. This is how learning works when it's easy. There's different things you might do, and you start here and you're not getting any reward because you don't know how to do it right. For example, how to extract the nectar from the flower. But you're trying out a few things, and these ones work a little bit better than those. So you stick with those, and then you try new things, and these ones work a little bit better. And you slowly climb the hill of getting better and better at extracting reward from the flower. And you can imagine this as being a general framework for learning in any context. But actually, difficult things might look like that, right? You initially do something, but nothing that's close to what you're doing initially even remotely works. So you're not getting any reward. Or even worse, what if it's like this? So if you're playing the piano and you're trying to impress your girlfriend, not playing the piano is probably sort of neutral. Playing the piano a little bit is probably bad, right? It just doesn't work. And if you want to get to here, you somehow have to survive that period, right? But what makes you survive that period? Now, if you're, I don't actually know what makes people play the piano, so I can't speak to that. But for bees, what could a bee do? If a bee is this little machine that's doing the thing that's most rewarded, it would never go through this valley, right? And so what I'm arguing here is that some data that Felicity Muth in my lab collected shows that actually innate bias can be the thing that pushes you through that valley. So how does anyone learn complex tasks? She had these artificial flowers that either had the nectar at the top or at the bottom of a kind of tulip-shaped flower where they had to go through some petals to get to the nectar well. And in this example, in this study, we use purple and pink flowers. And if the purple flowers are simple and the pink ones are complex, the bees never even bothered to land on the pink flowers. They literally never extracted nectar from a pink complex flower. And they did all their foraging on purple. I'm just showing the final trip, which is sort of the end conclusion that the bees arrived at. But it's true that they never extracted nectar from a pink complex flower. Now, if the purple flowers are complex and the pink ones are simple, they still go to a lot of pink flowers at the end. So a lot of bees still don't ever accept the complex purple flowers, but some at least visit both on the last trip. And if there are unequal rewards, in other words, the complex flowers have a much higher reward, then a good fraction of the bees learn the complex flowers if those complex flowers are purple. So the point that I'm making here is bees never learned complex flowers unless they were purple. So purple was what their bias led them to prefer in the beginning. And purple, and that is what allowed them to go through that negative phase of not extracting any rewards from any landings, but they were still persistent and trying because they liked the color so much. And that allowed them to, in the end, learn the complex flowers. So in this case, the initial bias, this prior, actually enables bees to overcome the reward valley in learning a complex task. Okay, so innate biases can be useful. Now, it looks like I have a scary amount of stuff there, but I'm actually just going to say this very briefly. And the brief thing that I want to say is all of this is very relevant also to how we think about life. So here's actually a final, not really data slide, but mentioning a study that we did. So David Kikuchi in my lab, in this case, had these different stimuli. And the way the study was set up was that in different conditions, there was a different amount of diversity in stimuli. In one case, there's just few similar stimuli. In another case, there's a larger amount of possible stimuli that the forager can choose from. And the question is, if in order to identify the best thing, you have to learn a combination of things. In other words, it's a yellow star. It's not blue stars. It's not everything that's yellow. You have to combine those two things to identify what the best thing is. Will the foragers learn that? And the fact of the matter is the foragers don't learn it if the diversity is high. Okay, does everybody get that so far? So diversity is high, foragers don't learn all the traits. But these foragers are humans. And so when you think about humans, I just want to read you this from the abstract because I think it's so relevant. We conducted experiments where humans acted as predators in a virtual ecosystem to ask how prey diversity affects the way that predators categorize prey phenotypes as profitable or unprofitable. The phenotypic diversity of prey communities strongly affected predator categorization. Higher diversity increased the likelihood that predators, people, would use a key trait to form broad categories, even if it meant committing errors. Now, we don't say this in the paper, but this is how stereotypes are born, right? This is what a stereotype is. It is, we just take one trait that categorizes something that's relevant, and we ignore all the other traits. We don't bother learning about the individuals. We just use this key trait to make decisions, even though they're often erroneous. Okay, does that make sense? So this applies to science and all kinds of areas of life. And so generally, I want to argue that we, like any other animal, are Bayesian reasoners. We do Bayesian reasoning. We combine our priors with our new information. And we have to be careful about that because we don't know where the priors come from. In a lot of cases, the priors are innate biases that are not, in some cases, not well studied in humans. Sometimes they are, but in a lot of cases may not apply to the current environment. And when we're doing science, basically the difference between these two approaches is that Bayesian reasoning is a great way to come up with best guesses, right? When we say something like unusual claims require unusual evidence, this is what we're talking about. Our priors are so against this claim that the new data has to be overwhelming in order to counteract our bias. And that is the right way to argue when we're arguing about broad things. But we have to remember that this is the method that allows us to ignore the priors. We can't really deliberately ignore our priors. This is how our brain is built. This is how a bee's brain is built. This is how any animal's brain is built. So we always have these priors when making any decision. And when you're doing a study, you have to be aware that this is what your brain wants to do. And so the reason that people argue this is what we should do in a scientific study is because we want scientific studies sometimes to give us answers independent of our priors. And this is why it's forcing us to have these alternative hypotheses and treat them the same, which is exactly not what we're doing there. Okay. Okay, so plenty of evidence shows that we humans have incredible power to avoid cognitive dissonance and bend any evidence to support the conclusion we want. This is another way of saying we have very strong priors about almost anything. And this is what strong inference is designed to avoid. So that method is designed to avoid us, arguing away all the evidence that disagrees with our priors. Okay, and so this is my second to last slide, I think, sorry. I'm not going to go into statistics today. I'm happy to answer questions about it if you want to ask me about it. But null hypothesis significance testing and p-values, there's sort of a discussion in the literature about p-values right now. And that's not the same as strong inference, but it is related to this. It's about having a clear yes or no answer where we can't somehow fudge it and we can't argue with it. We are forced to take a yes for a yes and a no for a no. That's what the purpose of having this significance testing is. When you do model fitting, when you have a whole bunch of models and you determine which one has the best fit, and you choose that as the conclusion from your study, you're doing Bayesian reasoning. That's the point of it. That's the point of model fitting. Okay. So ask about the why. I'll leave it at that. Think about the why. Think about your statistical methods. Think about your experimental design. And think about the rest of your life in terms of Bayesian reasoning and the fact that you're using priors to combine with current data. And bees are doing the same thing. Okay, and I'll stop there. And I'll just show my acknowledgement slide. Okay, I'm happy to take questions. Go back to this. What, I'm sorry, what? I can repeat the question, but you have to repeat it for me first.
You have to think about the why. Like what why exactly are you referring
to? Okay, so I guess I admit that the why, I'm leaving a lot of the thinking to you because I couldn't fit it into the talk anymore. But I guess what motivated me to make this link to the scientific method is that now in the literature, and I have this argument with my students and postdocs all the time about statistical analysis, a lot of times we abandon the idea of committing to our analysis before we have the data. So I think a lot of studies these days are not doing that. They're not committing to an analysis before they have data. And there are practical reasons for that that are hard to ignore. But what I'm saying is, if you do that, then you give up the rigor of this strong inference method. So the point of the strong inference method is before you roll the dice, you have to say exactly which roll will mean what. If you come up with that after you roll the dice, it's going to be heavily influenced by your priors. Let me give a concrete example. If I'm studying, let's say, what did we recently study? I mean, I'll give a very general example that's not to do with my work. You know, people study how brain size relates to sociality, right? And so the hypothesis is, if you live in larger groups, you should have a bigger brain because you need to deal with the social complexity. And it's a well-known criticism of that field that people don't actually say beforehand how they're going to analyze this. So when they analyze it, when, you know, now that you're sitting here and thinking about it, what do you think is the prediction from that hypothesis? Maybe the prediction is primates with larger group sizes have larger brains, but they don't actually. Okay, maybe the prediction is that they have larger brains relative to their body, but actually they don't. But maybe the prediction is that they have a larger cortex relative to the rest of their brain, and they do. So was that a good prediction for that hypothesis? I mean, if you'd said that a priori, that might have been a perfectly fine thing to argue. But the fact that you rolled the dice 3 times, right? You came up with something that was only your third choice among different possible predictions, means that the likelihood of any one of those correlations working was pretty high, and it wasn't 5%. Does that make sense? So if you just keep coming up with new predictions after you see the data until one of them fits your hypothesis, basically that is not what you're doing. And that's maybe something that's outside of what everybody here is doing. But you know, if you think about your own study, in ecological studies and behavioral studies, often there's a lot of things you could measure. And if we just measure all of them, and for example, now that we have more computing and more data, we include a lot of factors in our statistical models. And maybe when we planned the study, we thought we would only include factor A. But then we do the study and we think, oh, maybe we should also include factor B, and also the interaction between A and B, and then also C. And then also we should consider the day on which the study was done. And we start adding and adding and adding. We're really reducing the power of that method to prove ourselves wrong, because one of those versions will work eventually, no matter what the data look like. Molly.
Brendan who is attending remotely, has a number of questions, and I've asked him to prioritize. So he'll ask the other questions for you.
Feel free to email me with all your other questions.
So this probably should be our last one, but what he asks is, what would you predict if the consequences for a wrong decision are very severe, as in the high probability of death during a behavior? I assume he means exploration. What would you predict of the consequences?
Yeah, so I mean, we didn't try death. Actually, Shannon McWaters in my lab right now is studying predation, and so maybe we'll know the answer to that when it's literally death more. But in general, we do have, let's see here. So in a lot of these studies, we did actually change the cost of errors. And the question is actually a little more difficult than it at first appears, because there are errors. In a way, you could argue there are errors either way. Okay, this is a slide I didn't show, but I just want to explain that if you, basically there's the error that happens when you explore and we sample something that turns out not to be that good. So you explore something and it turns out what you explored was not a good thing. So now you know that, but you're not going back there. That's what we call a sample error. And an overrun error is where you fail to explore. You stick to the thing you know, but there's actually better stuff around. So that's really another type of error, right? And depending on what these two errors, how costly they are, you should explore more or less. So basically, if these errors are large, you should explore less. And if these errors are large, in other words, if the environment changes a lot or the variation between flowers is very high, so the really good ones have so much more nectar than the mediocre ones, then you should actually explore more to be more on top of what really is the best thing right now. So there's different types of errors, and I think the same, if I may still say that, the same applies to scientific studies, and I think it is not sufficiently addressed in our courses and discussions, societal discussions about how science should be done. So when you determine what the p-value is, for example, you're trading off between type 1 errors and type 2 errors. In other words, you're trading off between rejecting a correct null hypothesis and thinking the new thing is true versus rejecting a false a new thing and thinking the old thing is true, right? So those two things, if you're testing medication, right, is it better to approve a medication that doesn't actually work, or is it better to not approve a medication that does work? So in one case, you're failing to provide treatment when you could have, and in the other case, you're pretending to provide treatment, but it doesn't work. And which one of those is worse? It's not that easy to determine. It really depends on a case-by-case basis. You know, what are the alternatives? What are your chances of finding something else? How bad is the disease and all that stuff, right? And maybe in a lot of cases, we study things where we feel that the impact on direct human suffering is low, but really the costs of the errors are very important to consider in this whole discussion of how much to value new information versus old information.
Thank you, and thanks so much for your talk.